First of all, I would like to take this opportunity to thank the scientific committee for providing me a chance to share our findings in University Science Malaysia, all the way from Penang. And today I have the honor to talk about an aptamer that was generated against human pituitary tumor transforming gene 1, which is a global transcription factor. And I would like to share the diagnostic and the therapeutic potentialities of this aptamer. Okay, first I would like to speak briefly about the in vitro selection scheme of the aptamer against PTTG1, and I will move on to the diagnostics aspect of the aptamer, whereby I will cover the functionalization of the aptamer, the enzyme-linked aptasorbent assay that was developed, and I would also focus on the other two diagnostic assays that we have developed, which were aptamer-based dot plot assay / Western blot assay and aptahistostaining. And we are also quite excited to explore the therapeutic potentiality that can be offered by this aptamer. Therefore, we have carried out two simple assays to prove the therapeutic aspect of this aptamer. So let me give a brief introduction about PTTG1. PTTG1 is an intrinsically disordered protein, and it is involved in regulating sister chromatid separation, and it is also found to be involved in mitosis and cell cycle progression. And this transcription factor, being a global transcription factor, it is found to be overexpressed across various tumors. And the very high levels of PTTG1 correlates quite well with higher tumor grade, invasiveness, and tumor vascularity. And due to the presence of the protein across various tumor and its potential involvement in driving tumorigenesis, we suppose that having an aptamer would be able to eventuate into the diagnostic or therapeutic application of PTTG1. So therefore, we have actually cloned the gene that encodes for PTTG1. We have overexpressed and purified the protein. And as you can see from this figure, SPS page, both on native and denaturing page, we have actually acquired a very highly pure PTTG1. And we have also sought to confirm the identity of the protein by using monoclonal antibody using Western blot assay, and we realized that the protein that we have actually purified is indeed recombinant PTTG1. And having the protein in hand, we decided to embark on selects. So we use two different partitioning schemes in the selects, which are nitrocellulose-based partitioning and microtiter-based partitioning. And we have carried out up to 8 cycles of selects. And as I've mentioned previously, we use two different partitioning methods. The reason why we are using this is that when we alternate between different partitioning schemes, we realize that by doing so, it would be able to suppress the metric binder. So let's say we have binders that potentially bind to nitrocellulose filter, but subsequently, when you switch to microtiter-based partitioning, it would be able to suppress the propagation of the nitrocellulose filter binders. Therefore, by doing so, when we are able to suppress the metric binder, then this would actually assist in the enrichment of the potential binders. Besides that, we also increase the stringency by increasing the amount of competitors, as well as reducing the concentrations of both the RNA pool and the protein. And after 8 cycles of selects, we decided to monitor the enrichment. So we use a simple nitrocellulose filter binding assay, and we realized that our nucleic acid pool from cycle 8 has actually reached an enrichment of 28%. And this enrichment is approximately 9 times higher as compared to the nucleic acid from cycle 5 and cycle 0. So this affirms the fact that this nucleic acid pool from cycle 8 can potentially harbor the binders against PTTG1. So up next, what we did, we sent sequencing of approximately 50 clones. And upon the sequence analysis that we have carried out, we realized that there are three different classes of media sequences, while the rest of the sequences appear only once. And we decided to validate the binding of each and every single class of sequences by filter binding assay. And it turned out that this particular class by the name of Sekira 3 gave the best binding ability against the target protein, which were at 800 and 1600 nanomolar. And thus, we decided to choose this Sekira 3 as the potent aptamer against PTTG1. Now, having chosen the aptamer Sekira 3, we next decided to determine the KD or dissociation constant by using nitrocellulose filter binding assay. So we titrate the constant amount of aptamer against various concentrations of protein, and we arrive at a value of 16.41 nanomolar, which is in the low nanomolar range. And the next characterization that we have carried out is to determine the secondary structure of the Sekira 3 aptamer. And we have found out this aptamer forms sort of like an L shape, which have various types of loop. If we can take a look at the structure, we have internal loop, bulge loop, and hairpin loop. And besides determining the secondary structure of the aptamer, we have also decided to see whether this aptamer have distinct or overlapping binding sites on the surface of PTDG1 when we compare the binding with that of monoclonal antibody. So we have decided to assess this by carrying out a simple nitrocellulose filter binding assay. As you can see from this result, Sekira 3 aptamer was still able to bind to PTTG1 even in the presence of monoclonal antibody. So we hypothesize that what we can actually gather from here is the fact that even in the presence of monoclonal antibody, the binding of this aptamer towards PTTG1 is not perturbed. Therefore, we suppose that the binding site of Sekira 3 and monoclonal antibody are at two distinct sites on the surface of PTTG1 protein. And this is quite exciting because we can actually use both this aptamer and the antibody in concert in any sandwich assay. Okay, next, before we move on to the diagnostic aspect of Sekira 3 aptamer, we have to decide on the scheme of functionalization. How can we functionalize this aptamer with biotin for signal production? And what we have chosen is to rely on the duplex formation between the poly-A tail that is appended at the extremity of this aptamer with the biotin DT20. So it can be achieved by simple heating and cooling. So what we did, we just insert the sequence of this poly DA into the primer, and therefore after in vitro transcription, you would have a poly A tail appended at the end of this aptamer. And this poly A tail would then be able to form a duplex with DT20 that harbors a biotin at the end. And this biotin moiety would then be able to interact with the streptavidin moiety of HRP, which then can produce signal. So this is the functionalization strategy that we decided to adopt. And okay, after this functionalization, we really need to make sure whether the appendage of this poly-A tail at the end of the aptamer wouldn't interfere with the core secondary structure of this Sekira 3 aptamer. And with the aid of M-fold analysis, we realized that the appendage of this poly-A tail didn't really impair the secondary structure of Sekira 3. And we have also carried out a simple agarose gel shift assay to check for the duplex formation. If you take a look at this figure, agarose gel, at lane 2 particularly, we can see the band intensity is way higher than that of lane 1. And the higher intensity of the band is ascribable to the fact that poly-A tail is appended, or poly-A tail forms a duplex with the oligo-DT. So this extra strand would be able to incorporate more etherium bromide. So that's the reason that the band is way brighter than in lane 1. And we have also realized when we add streptavidin, there's a band that is retarded, And this is due to the interaction of this streptavidin with the biotin moiety. So this simple genship assay has actually successfully proven the fact that the biotinylated oligo DT is able to form a duplex with poly tail. So having done that, we can actually use this aptamer in the diagnostic platform. In terms of diagnostic platform, the easiest that we can actually access in our lab, or just like in any other labs, is actually in the form of ELISA assay. So what we have carried out 3 different types of enzyme-linked aptasorbent assay. The first one is the direct enzyme-linked aptamer assay, whereby we have actually immobilized PTDG1 directly by passive absorption, followed by the addition of aptamer and streptavidin HIP conjugate. The next scheme, we have adopted sandwich assay, whereby we use antibody as the capturing element, while aptamer acts as the detection agent. While in another scheme, we reverse the role of the aptamer. Let's just take a look at the result. So we have actually carried out select successfully, and we have achieved a limit of detection of 0.0256 with direct, while 3.2 and 16 nanomolar respectively for sandwich ELISA with aptamer as the capturing agent and sandwich ELISA with antibody as the capturing agent. There are differences in the detection limit here, and we suppose that this is due to the fact that PTDG1 as an intrinsically disordered protein, when it is being captured by the first capturing agent, and due to the conformational change imparted by the binding to the capturing agent, this probably reduces the binding of this PTDG1 with the detection agent. So that's the reason that there is a difference in terms of the limit of detection between sandwich ELISA and direct format. But the most important point that I would like to make here is the fact that Secura 3 aptamer has a promising potential on ELISA platform. And the next assay that we have carried out is apta dot blot assay and apta western blot assay. This is to see whether our PTDG1 aptamer is able to replace anti-PTDG1 monoclonal antibody in assays such as dot blot and western blot assay. And as you can see from this figure, it's clear that aptamer performs on par with that of antibody. And we are rest assured by this result that our aptamer have the potential to replace anti-PTG1 antibody in dot plot or western blot assay. And besides ELISA, dot plot, and western blot, we have also an interest in trying to see whether our developed aptamer is able to perform in histostaining application. And just to give a brief introduction about histostaining or immunohistochemistry, it is a process to characterize the intracellular proteins of various cell surfaces in all tissue. And it works based on the interaction of the antibody to the specific protein that is expressed on the surface of the cell. And due to the disadvantages of antibodies, such as batch-to-batch variation or high cost of production, we suppose that our aptamer or aptamers generally can actually replace antibodies. Thus, a new term is coined, which is known as apta-histostaining. So in this case, we want to see our Secura 3 aptamer is able to replace antibody in histostaining application. So we have carried out a simple histostaining using cell block, whereby the formalin fixed paraffin embedded cell blocks are made. Then these cell blocks are subjected to anti-generatable method using microwave, and this is followed by the addition of the blocking buffer, the biotinylated aptamer, streptavidin HRP, as well as staining with diaminobenzidine and counter staining with H and E. And we are quite excited to observe this result, as it shows that our aptamer has a higher staining capacity as compared to anti-PTDG1 monoclonal antibody. And this high degree of staining is not surprising, because aptamer, being smaller in size as compared to antibody, has a much deeper degree of penetration, and that would eventually translate into better staining. So it is safe to assert that Secura-3 aptamer is able to replace anti-PTTG1 antibody in histostaining application. Okay, we have shown that our aptamer harbors potential in diagnostic. We also have another question that ran in our mind back then. Can our aptamer block the interaction between PTTG1 and CXCR2, the interaction of which drives the tumorigenesis of tumor? In order to do that, so we have actually carried out a simple in vitro competitive nitrocellulose filter binding assay. So I will explain how it works. So what we did, we mixed Secura-3, PTTG1, and CXCR2 in a reaction. Then we subject to nitrocellulose filter binding assay. So PTTG1 being protein would be retained on the surface of the nitrocellulose membrane and together with its interacting partners. And the elute is then treated by RNase. And after the RNase treatment, agarose gel electrophoresis will then be carried out. So by doing so, we realize that when the concentration of aptamer increases, the amount of CXCR2 DNA fragment also increases in the elute. How does it happen? So the explanation here is that when the amount of aptamer is increased, the degree of inhibitory is increased as well. So aptamer is actually competing with CXCR2 to bind to PTTG1. So when this happens, when the amount increases, more unbound CXCR2 would then be collected in the elute. So that's the reason the intensity of the band that constitutes CXCR2 increases when the amount of aptamer increases. So coming back to the question, can Secura-3 block the interaction between PTTG1 and CXCR2? The answer is yes. And we have carried out another assay which is known as dual luciferase reporter assay. It uses two different plasmids, Firefly luciferase, which is a PGL3, and Renilla luciferase plasmid. And this assay hinges upon the fact that PTTG1 binds to CXCR2 in the vector. So when the binding happens, it drives the production of luciferase, which then carries the enzymatic action that eventually produce luminescence. So when aptamer is present, aptamer actually blocks the interaction between PTTG1 to the CXCR2 enhancer element on the plasmid. So when this happens, the production of luciferase is suppressed. Therefore, the signal luminescence will also be reduced. So this is how we're gonna see whether our aptamer is able to block the interaction between CXCR2 and PTTG1. And we realized when the concentration of this aptamer is increased, the fold changes, the signal production also reduces, as you can see from this figure, from 4 picomole to 32 picomole. So that means the inhibitory effect is actually a dose dependent. It happens in a dose dependent manner. So when you increase the amount of aptamer, it actually reduces the luminescence production as compared to the mock RNA. And this proves that the aptamer, Secura-3, is able to impart an inhibitory effect on PTTG1 transcriptional activity as compared to mock. And as a conclusion, we have successfully selected aptamer against PTTG1 with a KD value of 16.41 nanomolar. And this aptamer has demonstrated potentiality in the diagnostics, especially on ELISA, dot plot, aptamer-based Western blot, and hepatohistochemistry application. And besides in diagnostic application, this Secura-3 aptamer also demonstrated potentiality in therapeutic field in antagonizing the interaction between PTTG1 and CXCR2 enhancer sequence. And thank you so much for listening. Are there any questions?